David F. Stein, and hashtag X, David F. Stein 1, 2 Huidong Qian, and hashtag X, Huidong Qian 3, 4, 5, 6 Michael E. Vinyard, and hashtag X, Michael E. Vinyard 3, 4, 5, 6, 7 Qian, and hashtag X Qian 3, 4, 5, 6 Rebecca D. Combs, Rebecca D. Combs 4, 8 Zhang, 3, 4, 5, 6 Luca Pinello. Luca Pinello 3, 4, 5, 6. 1 Graduate School of Biomedical Sciences, Icahn School of Medicine at Mount Sinai, New York, United States. 2 Khoury College of Computer Sciences, Northeastern University, Boston, Massachusetts, United States. 3 Molecular Pathology Unit, Massachusetts General Hospital, Charlestown, Massachusetts, United States. 4 Center for Cancer Research, Massachusetts General Hospital, Charlestown, Massachusetts, United States. 5 Department of Pathology, Harvard Medical School, Boston, Massachusetts, United States. 6 Broad Institute of Harvard and MIT, Cambridge, Massachusetts, United States. 7 Department of Chemistry and Chemical Biology, Harvard University, Cambridge, Massachusetts, United States. 8 Windsor School, Boston, Massachusetts, United States. Single-cell assays have transformed our ability to model heterogeneity within cell populations. As these assays have advanced in their ability to measure various aspects of molecular processes in cells, computational methods to analyze and meaningfully visualize such data have required matched innovation. Independently, virtual reality, VR, has recently emerged as a powerful technology to dynamically explore complex data and shows promise for adaptation to challenges in single-cell data visualization. However, adopting VR for single-cell data visualization has thus far been hindered by expensive prerequisite hardware or advanced data pre-processing skills. To address current shortcomings, we present singlecellVR, a user-friendly web application for visualizing single-cell data, designed for cheap and easily available virtual reality hardware, for example, Google Cardboard, $8. SinglecellVR can visualize data from a variety of sequencing-based technologies including transcriptomic, epigenomic, and proteomic data as well as combinations thereof. Analysis modalities supported include approaches to clustering as well as trajectory inference and visualization of dynamical changes discovered through modeling RNA velocity. We provide a companion software package, singlecellVR, to streamline data conversion from the most widely adopted single-cell analysis tools as well as a growing database of pre-analyzed datasets to which users can contribute. 1. Introduction. Characterization of cell type, while once dominated by pathological description, has over the past decade shifted towards a more quantitative and molecular approach. As such, molecular measurements in single cells have emerged as the centerpiece of the current paradigm of mechanistic biological investigation, Trapnell, 2015. Technological advancements have enabled researchers to measure all aspects of the central dogma of molecular biology at the single-cell level, Stewart and Satija, 2019. Single-cell RNA sequencing, screen-a-seq, a technique that profiles the relative expression of genes in individual cells, and single-cell assay for transposis-accessible chromatin, using sequencing, scatic-seq, a technique that surveys genome-wide chromatin accessibility, are the most well-established and widely used of these methods. Buenrostro al., 2015, Loneman al., 2015. In fact, combined screen-a-seq plus scatic-seq assays are now routine, Perkel, 2021. Additionally, assays to profile DNA methylation, Luo et al., 2018, or protein levels are now maturing and becoming more widely accessible, Specht et al., 2019, Labib and Kelly, 2020. Most recently, combinations of various data modalities can now routinely be collected in parallel from the same cell, Chen S. et al., 2019, Zhu et al., 2019, Ma et al., 2020, Singh et al., 2020, Swanson et al., 2021. Screen-a-seq experiments generate on the order of millions of sequencing reads that sample the relative expression of approximately 20,000 to 30,000 transcribed features, for example, genes, in each cell of the sample. Normalized read counts for each feature can then be compared to discern differences between cells. Scatic-seq samples comprise a larger feature space wherein cells are characterized by the genomic coordinates of chromatin-accessible regions and sequence features derived from these regions, for example, transcription factor motifs, K-mer frequencies, etc. Initially performed in dozens to hundreds of cells, These experiments are now performed on the order of millions of cells, with a high-dimensional feature space as a result of thousands of features being considered for each cell and large, in cell number, experiments. Analysis methods for this data have been required to advance concurrently with the development of these technologies. Chen et al., 2019 c. Tian et al., 2019. With the exception of proof-of-concept methods still too nascent to be widely applied, Chen et al., 2021, omics measurements of single cells are generally destructive, preventing measurement of a cell at more than a single time point. As a result, most single-cell measurements for studying dynamic processes are of a snapshot nature, imposing inherent limitations on the study of such processes from this data. Weinrib et al., 2018. In light of this, transcription rates can be informative of ongoing processes in cells. The recent advent of RNA velocity quantifies and models the ratios of spliced and unspliced RNA, mRNA and pre-mRNA, respectively, such that they indicate the temporal derivative of gene expression patterns and thereby reflect dynamic cellular processes, allowing predictions of past and future cell states. Lamano et al., 2018. Among others, PCA, TSNE, and UMAP are dimensional reduction methods that have become common choices for enabling the visualization of high-dimensional single-cell datasets. Dimensionally reduced datasets are plotted such that similar cells cluster together and those with highly differing features are likewise clustered apart. In addition to the visualization and clustering of cells, trajectory inference methods have been proposed to learn a latent topological structure to reconstruct the putative time ordering, pseudotime, by which cells may progress along a dynamic biological process. Sellens et al., 2018. As single-cell technologies have advanced, techniques to cluster and organize cells based on single-cell assays have advanced alongside them, allowing key insights toward cell type and state characterization. Combined with RNA velocity information, trajectory inference can offer key insights on dynamical changes to cell states. Once in press, however, representation of these dimensionally reduced visualizations is limited to just 2 or 3 dimensions. Even using 3-dimensional plots from published studies, one cannot dynamically adjust or rotate the visualization to better understand the data from another angle. In addition, cells are typically annotated by features, for example time points, cell type or clusters, to investigate stratification along an axis of some biological process. To change the annotations presented in publication, one must often reprocess the raw data, which is time and skill intensive, highlighting the need for more dynamical visualization tools. While such current data representations are often limited and static, single-cell omic datasets are information-rich and, in many cases, important biological heterogeneity cannot be easily investigated or visualized outside the scope of the original publication, without spending considerable cost and time to reanalyze the datasets from scratch. VR visualization methods for single-cell data have been recently proposed. Yang et al., 2018, Legeth et al., 2019, Bresson et al., 2021. However, these methods require either expensive hardware or specific data inputs that mandate intermediate to advanced computational skills. Thus, tools and clear protocols are required to enable researchers, especially those who are not able to efficiently reprocess the raw data, to explore the richness of published datasets, or their own unpublished data, through a simple, easy, and affordable VR platform. Importantly, this platform must be flexible enough to accept all types of omics data from established and emerging technologies and processing tools currently employed by the single-cell community. At the time of this writing, 3 non-peer-reviewed methods employing VR technology that produce 2- and 3-dimensional visualizations of single-cell data have recently been reported. singlecellVR enables the visualization of standard SKRNA-seq data, though requires users to pre-process their data through scripting. Legeth et al., 2019. Unfortunately, this tool also requires expensive and dedicated VR hardware to operate. Another recent method for visualizing single-cell data in VR is Thea, Bresson et al., 2021, which has been designed with a focus on the exploration of spatial datasets for both RNA and protein measurements. Similar to singlecellVR, expensive computing power and VR hardware required to use Thea creates a barrier to entry. An alternative to these high-performance methods for VR visualization of single-cell data is StarMap, Yang et al., 2018, which allows the use of inexpensive cardboard visor hardware. However, StarMap lacks the advanced portability of outputs from commonly used SKRNA-seq analysis tools and limits cell annotation to clustering results of transcriptomic data. Of note, There are currently no peer-reviewed tools available for the visualization of single-cell data in VR, illustrating the novelty in this area of research. To overcome the limitations of these existing methods, as well as build on their qualities and initial progress, we present singlecellVR, an interactive web application, which implements a flexible, innovative visualization for various modalities of single-cell data built on VR technology. SinglecellVR supports clustering, trajectory inference, and abstract graph analysis for transcriptomic as well as epigenomic and proteomic single-cell data. Importantly, singlecellVR supports visualization of cell dynamics as described by RNA velocity, a recent milestone in the sequence-based analysis of single cells, Lamano et al., 2018, Bergen et al., 2020. SinglecellVR is a browser-contained, free, and open-access tool. Notably, we have developed a one-command conversion tool, SCVR, to directly prepare the results of commonly used single-cell analysis tools for visualization using singlecellVR. 2 results. 2.1 singlecellVR User Experience and Overview. SinglecellVR is an easy-to-use web platform and database that can be operated from inexpensive, cardboard visor hardware that costs as little as $8 and is available online from popular vendors including Google and Amazon. The webpage, available at http://www.singlecellVR.com, enables users to explore several preloaded datasets or upload their own datasets for VR visualization. Visualization can be done either on a personal computer or smartphone. To facilitate the transition between the personal computer browser view and the phone-enabled VR visor, VR mode, we have implemented an easy way to transition between these two visualizations as described in the next sections. In VR mode, an interactive visualization is presented to the user, allowing them to manipulate and visualize single-cell data using an array of annotations through the cardboard visor. Additionally, singlecellVR features the ability to receive as inputs the standard output files of commonly used tools for standard single-cell analysis, Sarah, Hao et al., 2021, Scampi, along with EpiScampi, Wolf et al., 2018, Denise et al., 2019, Stream, Chen et al., 2019a, Paga, Wolf et al., 2019, and SC Velo, Bergen et al., 2020. A companion package, SCVR, enables the conversion of these standard outputs to VR-compatible objects in a single command. In the sections below, we will describe the basis for this VR visualization platform as well as provide descriptive examples of the visualization that can be performed using singlecellVR. We will compare singlecellVR to existing methods and describe its unique advantages that build on the early progress of single-cell data visualization in VR. We include a detailed protocol and quick start guide that describe how the web platform enables researchers to explore their own data and dually functions as a database for pre-formatted datasets that can be explored immediately in VR. Supplementary Note 1. 2.2 VR Database and SCVR Pre-Processing Tool. SinglecellVR provides a growing database of several datasets processed for VR visualization. Initialization and future growth of this database is enabled, scale-free through the streamlined SCVR utility. As shown in Figure 1, to use singlecellVR, the user may select a pre-computed dataset or convert their data from commonly used single-cell workflows. This conversion can be easily accomplished by using SCVR, a simple one-line command tool for performing data conversion, and produces a simple zipped JSON file with all the information required for visualizing cells and their annotations in VR. Additionally, datasets for which RNA velocity information has been calculated may be submitted directly for visualization of velocity in VR without prior conversion. Supplementary Note 2, Supplementary Notebook 4. Figure 1. www.frontierson.org. Figure 1. An overview of the singlecellVR user experience. Top, gray, the outputs of a standard two-dimensional screen-a-sec analysis. Middle and bottom, purple, a step-by-step overview of the singlecellVR workflow, one schematic of flexible data conversion, one command to install, via the Python pip package manager, and one command to convert the data to be VR compatible. 2. Web page for uploading and exploring VR data. 3. VR mode visualization using a cheap smartphone-enabled headset. Conversion from the standard output of any single-cell analysis tool to this format would normally pose a significant methodological roadblock to most users, especially non-computational biologists. To bridge this gap, SCVR parses and converts the outputs of Scampi, EpiScampi, Sura, Paga, and Stream, respectively. Loom H5 add and PKL, and creates the required zipped JSON file. Supplementary Note 2. This file contains the 3D coordinates of cells in a specified space, for example UME, LLE, etc., cell annotations, for example fact sorting labels, clustering solutions, sampling time or pseudotime, etc., and feature quantification, gene expression levels, transcription factor deviation, etc. It also contains the graph structure, the coordinates of nodes and edges, obtained from supported trajectory inference methods. Users interested in visualizing skirnosec dynamics using RNA velocity generated from spliced and unspliced read counts can likewise prepare this information for visualization in singlecellVR using the singlecellVR companion utility. Users can follow established workflows for obtaining these insights from the raw read file inputs, as well as make use of the tutorials available at the singlecellVR GitHub repository, Section 5, Supplementary Notebook 4. Importantly, singlecellVR has been made available as a Python pip package to streamline its installation and can convert a process dataset for VR visualization with a simple command. To install, one can simply open their command line utility and run pip install singlecellVR. Once installation is completed, the user can navigate to https://github.com/pinellolab/singlecellVR to copy and customize the example commands provided to execute the one-step process for converting their data to a VR-compatible format. In addition to the documentation of singlecellVR, we have filmed a short video tutorial found on the homepage of singlecellVR to further assist less experienced users in preparing their data for visualization. To showcase the functionality and generalizability of singlecellVR across data types, we have pre-processed a collection of 17 published datasets, which includes both skirnosec as well as scatic-sec and single-cell proteomic data, and made them available for immediate VR visualization. Taken together, we believe this step addresses a key limitation of previously developed VR tools mentioned above, and a formal comparison is presented in Section 2.5, Yang et al., 2018, Legeth et al., 2019, Brasson et al., 2021. Excitingly, given the small footprint of the files obtained with singlecellVR, we are offering users the ability to easily submit their process data to the singlecellVR GitHub repository, see Supplementary Figure S1, to make the tool a general resource for the field. In this way, we hope to even further extend the ability of biologists to visualize once static datasets and easily generate new hypotheses through manipulation of a large number of rich datasets. Therefore, we envision that our website will function as a repository for VR visualization data of single-cell biological annotations. 2.3 A simple, cloud-based web tool for VR visualization. singlecellVR is available as a web app at http://www.singlecellVR.com. This website enables users to explore several preloaded datasets or upload their own datasets for VR visualization. To build singlecellVR, we have adopted recent web technologies, Dash by Plotly and A-Frame, a recently developed JavaScript framework for VR/R. This allowed us to create a tool that is portable and does not require any installation. The website can be reached through any web browser, and browser compatibility was tested against Google Chrome, Apple Safari, and Mozilla Firefox. Visualization can be done either on a personal computer or smartphone, both Android and Apple smartphones. Once the users have uploaded their data to singlecellVR, they have the option to view and explore the data in 3D, directly in their web browser, or to quickly jettison the data to their mobile device for visualization in a VR headset, Figure 2 and Supplementary Figure S2. A key challenge associated with developing a method for visualization of single-cell data is transporting data that is typically processed in desktop settings to the smartphone-based VR visualization. In fact, we predict that in most cases, users will prefer to upload their data through a computer in which they may have run their analyses. To overcome this challenge and enable a seamless transition to a smartphone for VR view, our website dynamically generates a QR code that enables users to open the VR view on their phone to view data uploaded through a personal computer. This mixed approach is particularly useful because, as mentioned before, most users are not processing single-cell data analysis from a phone, nor would they keep the files on a mobile device. Figure 2. www.frontierson.org. Figure 2. Step-by-step protocol for data processing and using singlecellVR. Step 1. Single-cell data can be generated using a variety of technologies or downloaded from online repositories. Data can then be pre-processed and prepared for use, most often as a feature matrix, with common single-cell analysis tools. Step 2. Pre-processed data can be analyzed using common single-cell analysis tools, listed here. Step 3. Users can process their data for use with singlecellVR from any of the standard outputs created by analysis tools listed in Step 2. Listed at the top in a single command. Step 4. Users can select from pre-processed data or upload their own data, Step 4b, and scan the dynamically generated QR code with their phone to begin the VR visualization, Step 4c. Step 5. Users can use the QR code on the website to transfer their data to their phone for use with simple hardware, and are explained in the Section 5 section. Step 1. 2.4 Supported Tools and Analysis. 2.4.1 Visualizing Single-Cell Clustering Solutions in VR. As previously mentioned, Scampi and Seurat are two commonly used tools for performing cell clustering as well as differential expression analysis. Here we demonstrate the utility of singlecellVR to visualize the common outputs of these tools, showcasing both the clustering solutions as well as differentially expressed genes or other technical or biological features that are visualized easily through the VR interface. Figure 3. A key advantage of our tool is the ability to supply multiple annotations to cells to visualize various attributes of the measured data, for example, based on a biological query of interest or experimental design. This may include stratification by cluster identity, time points, tissues, or facts-based labels. In Figure 3, we demonstrate the ability to select visualizations by various cluster identifications, which are user-customizable. With the advent of cross-experiment integration methods that can integrate not only multiple skRNA-seq experiments but experiments across modalities of single-cell data collection, this flexible labeling strategy should enable the user in the future to visualize even the most novel and complicated experiments in rich detail. Figure 3. www.frontierson.org. Figure 3. VR visualization of single-cell process datasets profiled by different technologies and analyzed by various computational tools. A. Scampi offers solutions for clustering single-cell data. Shown is a map of the Allen Brain Atlas Mouse Brain skRNA-seq dataset from Yao, 2020, and processed by Scampi. Leiden clustering solution, left, and an expression of GAD1, right. B. Trajectory inference applications. PAGA offers a partition-based graph abstraction to uncover potential trajectories, edges, between group of cells, nodes, top left, relative gene expression, for example, KLF1, top right, amongst other annotations. The PAGA analyzed dataset shown here is from Paul et al., 2015. Stream offers the visualization of developmental trajectories, which can be visualized by cell identity, bottom left, or by relative gene expression, for example, GATA1, bottom right, amongst other annotations. The stream analyzed dataset shown here is from Nastoroa et al., 2016. C. Epigenomic applications. Scampi enables the clustering and visualization of scatic-seq data, left. PBMC, healthy donor, 10,000 cells dataset analyzed by Scampi and with colors corresponding to clustering solutions, Louvain clustering. Stream was used to perform trajectory inference on scatic-seq dataset when Rostro et al. 2018, right. D. Seurat offers solutions for clustering single-cell data as well as integrating datasets across experiments. Shown is a Seurat integrated skRNA-seq and scatic-seq PBMC dataset from 10x Genomics, colored by technology, left, and cell type, right. In addition to flexibility for visualizing complex experimental setups, singlecellVR is able to visualize large experiments. To demonstrate this utility, we first processed, using Scampi and SCVR, and visualized on singlecellVR, skRNA-seq data from the Chan Zuckerberg Biohub Tabula Muris project, a dataset consisting of 44,949 cells and 20 tissues from 7 mice, Shom et al., 2018. In supplementary Figure C, clustering analyses of this dataset are projected into VR, colored by mouse tissue, left, and Louvain cluster identity, right. With a quick rendering time, less than 1s, for the Tabula Muris dataset, we next explored the realm of visualization for a modern, large atlas-scale dataset, greater than 1m cells. Using Scampi and SCVR, we successfully processed and visualized on our website cells from the Allen Brain Institute that capture cortical and hippocampal development inside the mouse brain. Figure 3a, Yao, 2020. This dataset consists of 1,093,785 cells and is among the largest screenosec datasets created to date. Visualization of this dataset in a dynamic VR setting creates the opportunity for more in-depth study of subsections of the data, which is particularly valuable for such a large dataset. While the datasets visualized in this manuscript were obtained in their pre-processed state, we have created IPython notebook tutorials for integrating datasets from multiple transcriptomic experiments as is performed in Seurat. These may be accessed in the associated GitHub repository. 2.4.2 Visualizing Single-Cell Trajectory Inference Results in VR. Single-cell measurements are particularly useful for capturing cross-section snapshots of a biological process. With dense cell sampling, one can often observe transient cell states that exist between two more stable states. However, without an intrinsic understanding of the process being studied, it may be difficult to order these cells along a time axis of a biological process. To enable ordering cells by transcriptional, or epigenomic, states, pseudotemporal ordering, based on trajectory inference and machine learning algorithms has become a useful technique for the single-cell field. Trajectory inference, like clustering, describes a high-dimensional biological process, and being limited to a 2/3-dimensional static visualization on paper, with a limited selection of genes or annotations is not ideal. Thus, we intend for our tool to leverage the richness of these datasets and make their general usefulness to the field more widespread. We therefore wanted to extend our VR visualization to the results of common trajectory inference tools, Figure 3b. singlecellVR supports two trajectory inference tools, PAGA, a partition-based graph abstraction trajectory inference method, and STREAM, a method based on principal graphs, Albergante et al., 2020. That recovers a tree-like structure to summarize developmental trajectories and to visualize the relative densities of cell populations along each branch. To showcase the ability of singlecellVR to visualize trajectory inference results, we reprocessed a popular myeloid and erythroid differentiation dataset, Paul et al., 2015, performing trajectory inference using PAGA. PAGA is designed specifically to preserve relative cell topology in constructing the trajectory along a pseudo-time axis. In the depiction of the PAGA-generated trajectory, nodes, gray, correspond to cell groups, and edges, black lines between nodes, connecting the groups quantify their connectivity and confidence, thickness, Figure 3b, top. To showcase the VR output of STREAM, we reprocessed a popular mouse blood dataset, Nestaroa et al., 2016. In STREAM, a set of smooth curves, termed principal graph, are fitted to the data and each curve represents a developmental branch. Within singlecellVR, we are able to easily explore these trajectories and observe qualitatively the distribution of cells along each branch in the UMAP space, Figure 3b, bottom. The branches of these trajectories are represented by the curves that cut through the cells. SinglecellVR and SCVR also support processing and visualizing single-cell epigenomic data. To demonstrate this functionality, we first used the EpiScampi workflow to cluster a scatic-seq dataset from 10x genomics containing 10,000 cell PBMC, healthy donor, Figure 3c, left. Next we reprocess with STREAM a scatic-seq dataset profiling human hematopoiesis, Rostro et al., 2018, Figure 3c, right. In addition, we extend singlecellVR to single-cell quantitative proteomics data. To this end we reprocess data from Scope 2, a recent assay to quantitate proteins in single cells using mass spectrometry, Specht et al., 2019. We perform trajectory inference using STREAM on one Scope 2 dataset profiling the transition from monocytes to macrophages in the absence of polarizing cytokines. Our analysis revealed a bifurcated branch structure as cells progress towards macrophage phenotypes, supplementary Figure Seb. Importantly, such bifurcation is not readily visualized in previous reports in 2 dimensions. Finally, we took advantage of the recent advances in the multi-omics field, using Seurat to integrate and co-embed PBMC cells profiled by skRNA-seq and scatic-seq by 10x genomics, Figure 3d. 2.4.3 Visualizing RNA velocity analysis in VR. Having successfully applied the singlecellVR framework to the visualization of trajectory inference analyses for multiple modalities of single-cell data, we sought to extend the framework further to visualize dynamical changes at single-cell resolution by way of RNA velocity. We first demonstrated this on a popular endocrine pancreas dataset, Figure 4, which has been previously employed to demonstrate the utility of visualizing dynamic processes using velocity. Figure 4. www.frontierson.org. Figure 4. VR visualization of single-cell datasets with RNA velocity. SC-Velo enables efficient analysis of the RNA velocity attributes of single-cell data. Shown is a 3D map of an endocrine pancreas dataset, Bastidas Ponce et al., 2019. A. Cells are displayed as their corresponding 3D velocity vectors and colored according to cluster annotation. B. Cells are displayed as 3D orbs surrounded by a corresponding grid of velocity vectors. Cells are colored according to cluster annotation. Visualization of RNA velocity using singlecellVR has two modes. In the default mode, each cell is represented by an arrow where the magnitude and direction of the arrow denote the velocity of that cell. Figure 4a. For larger datasets, cells may be represented as spheres, while a surrounding grid system of arrows denotes the predicted trajectory of a given cell. Figure 4b. This is particularly helpful for interpreting the overall direction of cells in various clustering regions or subsets of a given trajectory. In either mode, the arrows are animated to gravitate towards the direction of the corresponding cell trajectory. Latent time, t, is a parameter of the velocity calculation for a given cell. To aid in user comprehension of observed velocity, the speed and distance of the animated velocity vector may be calibrated on the fly during the VR experience through adjustment of the t parameter using the floating VR assistance menu. These results, taken together with the visualizations of clustering analyses as well as trajectory inference analyses, indicate that singlecellVR is a robust, generalizable tool across multiple modalities of single-cell analysis. 2.4.4 Creating reproducible visualizations. To enable singlecellVR users to create visualizations that can be reproduced upon sharing, we have included a feature in the VR interface, which captures absolute x, y, and z coordinates such that one may navigate to an identical position with precision. In line with this, we have also included pitch, yaw, and roll descriptions of the camera angle view. These position descriptions of the VR viewpoint can be captured and shared as part of the visualization. Viewpoint descriptions may also be toggled on or off. Supplementary figure S4. 2.5 Comparison of singlecellVR to existing methods. As mentioned above, there are currently 3 unpublished reports of VR tools created to visualize single-cell data: singlecellVR, Legit et al., 2019, StarMap, Yang et al., 2018, and Thea, Brisson et al., 2021. In this section, we compare these tools to singlecellVR on 2 axes: 1. Ease of use, and 2. Overall performance for visualization and analysis in VR. 2.5.1 Ease of use. Competing tools. Both singlecellVR and Thea require or recommend HTC Vive or HTC Vive Pro VR hardware, $500 to $1,000, an Intel Core i7 processor, $300, or better, an NVIDIA GTX 1080 or NVIDIA GeForce RTX 3080-3090, $1,500 to $3,000, 16 to 32 GB RAM, $50 to $150, and a solid-state hard drive, SSD 1 TB SSD recommended for singlecellVR, $50 to $100. Altogether, this equipment requires a minimum investment of roughly $2,300 $4,550. These are computational equipment that most biologists will not have at their disposal within their lab, likely limiting use of this tool to more computationally focused labs. singlecellVR requires software and boilerplate level to pre-process the data in preparation for VR visualization is required, and therefore requires the user to perform scripting to prepare data for downstream use with the VR visualization. While Thea has provided a convenient Python script to convert AND data objects, their software is not open source, hindering further community contribution. A contrasting alternative to singlecellVR and Thea is StarMap, which is compatible with low-cost hardware such as Google Cardboard. However, StarMap takes as input separated values containing information of the 3-dimensional coordinates of cells in the visualization as well as annotations, for example, cluster ID, and up to 12 features per cell. This file must be prepared entirely by the user without assistance from the StarMap platform, limiting the audience of this tool to experienced computational biologists. SinglecellVR. The Single Command Companion Package for Data Preparation, SCVR, described above enables users to visualize their own pre-computed data directly from the outputs of commonly used single-cell RNA-seq analysis tools. Currently supported tools include ScanPy, EpiScanPy, Sura, Paga, Stream, and SC-Velo. SinglecellVR is the only tool of the three discussed, singlecellVR, Theia, and StarMap, that features a QR code to quickly transport the VR data visualization to another device. 2.5.2 VR Performance and Analysis Capabilities. Competing tools. singlecellVR proposes a versatile, user-friendly visualization for standard SCRNA-seq workflow outputs and demonstrates comparable utility on scatic-seq data. Theia offers a similarly high-performance visualization of single-cell data. Theia's key distinguishing contribution is its visualization of spatial transcriptomic single-cell datasets. StarMap is only demonstrated on SCRNA-seq data and lacks the ability to visualize analyses beyond clustering, such as trajectory inference or an illustration of velocity. Further, StarMap is only capable of displaying up to 12 features for a given cell, limiting the throughput with which users may analyze their data. SinglecellVR. In contrast to existing methods, SinglecellVR offers both a high-performance visualization with in-depth analysis and the ability to visualize all modalities of data at scale, while at the same time offering a software that is compatible with low-cost hardware and requires minimal computational abilities. These advances, which build on the progress made by these initial methods, create a tool which offers a low-cost alternative to existing tools with virtually zero barrier to entry, while maintaining high-performance VR visualizations. 3. Discussion. The amount of publicly available SCRNA-seq data has exploded in recent years. With new assays to capture chromatin accessibility, DNA methylation and protein levels in single cells, we predict a second wave of dataset generation. Each of these datasets is extremely high-dimensional and thus, rich with latent information about a given biological sample. Ideally, biologists would be able to explore this treasure trove of data from any angle and make hypotheses assisted by in silico analysis at little to no time cost. Often, however, experimental biologists lack the advanced computational skills and/or time required to reprocess and reanalyze raw data from published experiments to gain an understanding of the data from their desired angle of interest. Additionally, biologists who wish to thoroughly explore data prior to publication may rely on a computational specialist who is less connected to the biological problem of interest, introducing a disconnect in hypothesis-driven experimental turnover. While once primarily reserved for entertainment, VR has found utility in both industrial and academic applications. In this manuscript, we present a protocol for visualizing single-cell data in VR. This protocol is based on SinglecellVR, a VR-based visualization platform for single-cell data, and discusses its innovations and differences with existing methods. Importantly, we provide a simple mechanism to prepare results from commonly used single-cell analysis tools for VR visualization with a single command to considerably increase accessibility. See Section 5. With this added utility, we seek to empower non-computational biologists to explore their data and employ rapid hypothesis testing that could not be made from the traditional static representations typical of communication in a scientific report on paper or a computer screen. We anticipate that VR will become increasingly useful as a research and education tool, and that the construction of software libraries will aid such advancements. VR has also recently found application in other sources of biological data, including single neuron morphological imaging data, Wang et al., 2019, three-dimensional confocal microscopy data for fluorescent molecule localization, i.e., fluorophore-tagged proteins, within cells, Stefani et al., 2018, and three-dimensional single-molecule localization super-resolution microscopy, Spark et al., 2020. Our scalable and flexible VR visualization framework is not limited to SCRNA-seq, and it can be also easily adapted to other single-cell assays and tools that already support epigenomic data and/or single-cell proteomic data, EpiScanPy, Denise et al., 2019, Sura, Stewart et al., 2019, and Stream, Chen et al., 2019a. Finally, we extend our framework to computational methods that derive the RNA velocity of single cells for visualization in VR, Lamano et al., 2018, Bergen et al., 2020. With the recent advances in spatially resolved transcriptomics, Welch et al., 2019, and corresponding analysis methods, Howe et al., 2021, Miller et al., 2021, visualization of such data has already been extended to a VR framework, Brassen et al., 2021. We believe this new sort of three-dimensional VR will also become especially useful once made available to the general research community via inexpensive hardware and facile data pre-processing and preparation for VR visualization. As software to analyze single cells reach their maturity, one could imagine the incorporation of such visualizations into more clinically translatable settings, such as medical devices. For conclusion, this manuscript presents singlecellVR, a scalable web platform for the VR visualization of single-cell data and its associated pre-processing software, SCVR, which streamlines the results of commonly used single-cell workflows for visualization in VR. SinglecellVR enables any researcher to easily visualize single-cell data in VR. The platform is user-friendly, requires no advanced technical skills or dedicated hardware. Importantly, we have curated and pre-processed several recent single-cell datasets from key studies across various modalities of data generation and analysis approaches, providing the scientific community with an important resource from which they may readily explore and extract biological insight. 4.1 Key Points. SinglecellVR is a web platform that enables quick and easy visualization of single-cell data in virtual reality. This is highlighted by a database of preloaded datasets ready for exploration at a single click or via a QR code to quickly jettison the visualization to a smartphone-enabled VR visor. SCVR is a companion package to easily convert standard outputs of common single-cell tools in a single command. SinglecellVR is made for use with cheap and easily available VR hardware such as Google Cardboard, $8. SinglecellVR can visualize both clustering solutions as well as trajectory inference models of single-cell data for transcriptomic, epigenomic, and proteomic data, as well as multimodally integrated datasets. Additionally, SinglecellVR offers a three-dimensional VR visualization of RNA velocity dynamics. 5 Materials and Methods. 5.1 Single-cell data preparation. All datasets were processed using SCAMP, version 1.5.1. Red SCR_018139, and data, version 0.7.6, Red SCR_018209, EpiSCAMP version 0.1.8, Sura version 3.1.5, Red SCR_007322, Paga, part of SCAMP, version 1.5.1, Red SCR_018139, Stream, version 1.0, and SC Velo, version 0.2.3, Red SCR_018168, following their documentations. Jupyter notebooks to reproduce data processing are available at https://github.com/pinellolab/singlecellVR. Analyses were performed on a 2019 MacBook Pro, 2.4 GHz, Intel Core i9, 16 GB RAM. 5.2 Preparation of processed data for visualization in VR. The pre-processing package, SCVR, generates a series of JSON files containing the spatial coordinates representative of cell embeddings in 3D embedding, for example, PCA, UMAP, etc., and information including labels and features, for example, gene expression, TF motif, deviation, etc. These JSON files are zipped upon output from SCVR into a single file that can be easily uploaded to SinglecellVR for visualization. 5.3 Singlecellvr Web App Construction. To build Singlecellvr, we used a frame, version 1.2.0, dash by Plotly, version 1.13.3. Data Availability Statement. The source code and the supporting data for this study are available online on GitHub at https://github.com/pinellolab/singlecellVR. The pre-processing package, SCVR, is included within that repository https://pippy.org/project/SCVR/. The documentation for SCVR is available here. https://github.com/pinellolab/singlecellVR. Video tutorials for learning about and running visualization experiments with Singlecellvr and using SCVR to prepare the data are available on YouTube, here. https://www.youtube.com/playlist?list equals plxqlnt g-kilb ma a ibston bzun 6a ulix 8. All the analyses in this article can be reproduced using the Jupyter notebooks available at https://github.com/pinellolab/singlecellVR. Additionally, we have provided a wiki within the same repository for a more detailed guide to reproducing results from the paper as they pertain to the supplementary materials. Author contributions. Authors DFS, HC, MEV, and QQ contributed equally to this publication. DFS, HC, MEV, and LP conceived this project and designed the experiment, which was begun at the 2019 HackSec at the University of British Columbia, where input from collaborators mentioned in the acknowledgements was received. DFS, HC, MEV, and RDC processed the data hosted in the database as well as produced the VR image demonstrations of singlecellVR shown in this manuscript. DFS led the development of the virtual reality framework. HC led the development of Dash-based website and the pre-processing module, SCVR. QQ contributed to extending the VR tool with velocity and new API. QZ contributed to extending the VR tool to incorporate single-cell protein analysis. MEV led the preparation of the manuscript. All authors performed user testing of the software. LP supervised the development of this work and provided guidance. All authors wrote and approved the final manuscript. Funding. This project has been made possible in part by grant number 2019 to 202,669 from the Chan Zuckerberg Foundation. LP is also partially supported by the National Human Genome Research Institute, Khoury, Career Development Award, ROOHGOO8399, and Genomic Innovator Award, R35HG010717. MEV is supported by the National Cancer Institute, NCI, Ruth L. Kirstein, NRSA Individual Predoctoral Fellowship, 1F31 Ka257625001. Conflict of interest. The authors declare that the research was conducted in the absence of any commercial or financial relationships that could be construed as a potential conflict of interest. Publisher's note. All claims expressed in this article are solely those of the authors and do not necessarily represent those of their affiliated organizations, or those of the publisher, the editors, and the reviewers. Any product that may be evaluated in this article, or claim that may be made by its manufacturer, is not guaranteed or endorsed by the publisher. Acknowledgements. We would like to acknowledge the organizers and participants of HackSec 19, University of British Columbia, where this project began. We would like to especially acknowledge those HackSec 19 participants that contributed to the development of this project: Michelle Crown, Alexander Dungate, David Lin, Terry Lin, and Sepan Dayanakar Moded. Additionally, we would like to thank Ashley Brown, Sulma Ibrahim, and Kate McCurley for their contributions to the construction of the singlecellVR database through the Windsor Science Internship Program. Supplementary material. The supplementary material for this article can be found online at https://www.frontierson.org/articles/10.3389/FEGene.2021.76417/0/full #supplementary material. Supplementary Figure 1. Instructions for contributing VR process data to the singlecellVR data repository. Users can contribute to the growing repository of VR datasets by submitting a pull request to our GitHub repository, https://github.com/pinellolab/singlecellVR. To do so, first fork and clone the repository, steps 1 and 2, above. Next, add your data, step 3. Finally, create a pull request, step 4, to be submitted for approval. Once approved, your data will be incorporated into the growing repository of VR datasets. It is necessary to add the VR dataset flag, purple, already added to the sidebar, to the pull request. In addition, we ask users to describe the data, methods used, and available annotations, for example genes, time points, clusters, labels, etc., in the commit message or comment section of the pull request. Note: for velocity results, files greater than 50 megabytes are too large to be shared through GitHub and must be shared via other channels. However, coordination of this sharing may proceed through GitHub as shown in this figure. For more, see Supplementary Note 2 and Supplementary Notebook 4. Supplementary Figure 2. Tips for using the VR interface. A. It is not required, but one can easily connect a keyboard with your smartphone using a Bluetooth-enabled keyboard. A small portable keyboard can be purchased from Amazon for $10. However, you can still use a normal computer with your browser and explore using your mouse and keyboard, the three-dimensional transcriptional space with cells, trajectories, and graph abstractions. The full set of interactive keyboard functionalities are detailed above. B. There are several similar versions of cardboard VR adapters available for $8. Many VR headsets such as Google Cardboard have a single button that allows a user to click the screen of their phone while immersed in a virtual reality experience. By holding down this button, users without a keyboard may move forward in the direction of their gaze. You can also simply use your computer screen to do initial exploration of the data in 2D. Users may navigate the VR visualization via a combination of gaze controls and keyboard inputs. A circle, centered in the user's field of view, indicates the direction that a user will move through the virtual space and also acts as the appendage through which the user will interact with objects in the visualization. Additionally, users may select the keyboard button on the menu to render a virtual keyboard. Cardboard users may use this keyboard to search for available features to render on the display. The Enter/Return key on the virtual keyboard clears the current search. Subsequently, selecting the keyboard button will hide the keyboard from view. Supplemental Figure 3a. Rendering the single-cell virtual reality visualization. Scampi offers tools for clustering, which can be visualized using singlecellVR. Cells can be visualized and colored by various annotations. Shown, mouse tissue type, left, or their cluster ID, right. The Scampi analyzed dataset shown here is from the Chan Zuckerberg Initiative's Tabula Murray's dataset, Shom et al., 2018. B. Stream process single-cell proteomics data from Scope 2, Specht et al., 2019. These visualizations are an example of an advantage gained by trajectory analysis and three-dimensional visualization. 3D UMAP plots, ordered left to right, top to bottom, generated by stream, respectively colored by pseudotime progression, cell type, orange, monocyte, blue, macrophage, expression of SafeAbe, and expression of PFN1. Supplementary Figure 4. Camera coordinates and angle descriptions enable reproducible visualizations. Shown is a UMAP of the Allen Brain Atlas Mouse Brain Screen Sec dataset Yao et al., 2020, Yao, 2020, and processed by Scampi colored by the Leiden clustering solution. Close-ups of the coordinates as well as the toggle for displaying coordinates are shown. References: Alberganti, L., Merckx, E., Bach, J., Chen, H., Martin, A., Foray, L., et al., 2020. Robust and Scalable Learning of Complex Intrinsic Dataset Geometry via LPyGraph. Entropy 22. 296. DOI 10.3390/E22030296. Bastidas Ponce, A., Trichler, S., Doney, L., Scheibner, K., Tarkas Medina, M., Solno, C., et al., 2019. David F. Stein, and hashtag X, David F. Stein 1,2 Huidong Chen, and hashtag X, Huidong Chen 3,4, 5, 6 Michael E. Vinyard, and hashtag X, Michael E. Vinyard 3,4, 5, 6, 7 Chen Chin, and hashtag X Chen Chen 3,4, 5, 6 Rebecca D. Combs, Rebecca D. Combs 4, 8 Qian 19, three-dimensional confocal microscopy data for fluorescent molecule localization, i.e., fluorophore-tagged proteins, within cells, local coordinates representative of cell embeddings in 3D embedding, for example, PCA, UMAP, etc., and information of VR datasets. It is necessary to add 020-0591-3. Brasson, D., Mulvey, C., M., Kosaj, F., Becker, R., Grimaldi, F., Coffey, S., et al., 2021. Exploration and analysis of molecularly annotated, 3D models of breast cancer at single-cell resolution using virtual reality. BioArXiv 06, 448342. DOI 10.110.12021.06.28.448342. Buenrostro, J.D., Courses, M. R. Leroux, C. A. Wu, B. Shep, A. N. R.E. M. J. et al., 2018. Integrated Single Regulation. Cell 173, 1535-1540. J.Cell.2018.03.074. Buenrostro, J. D. Wu, B. Lisenberger, U. M. Ruff, D. Gonzalez, M. L. Snyder, M. P. et al., 2015. Single Cell Chromatin Accessibility Reveals Principles of Regulatory Variation. Nature 523, 486-490. DOI 10.1038/Nature 14590. Chen, H. Albergante, L. Shu, J. Y. Leroux, C. A. Lobosco, G. Guan, J. et al. 2019 A. Single cell trajectories reconstruction, exploration and mapping of onyx data with stream, NAT, command 10. DOI 10.1038/S414670190967004. Chen, H. Leroux, C. Andrew T. Vinyard, M. E. Garcia, S. P. Clement, K. et al. 2019 C. Assessment of computational methods for the analysis of single cell ADAC-SEC data. Genome Biol, 20. DOI 10.1186/S13059-019-1854-5. Chen, S. Lake, B. B. and Zhang, K. 2019 D. High throughput sequencing of the transcriptome and chromatin accessibility in the same cell, NAT, Biotechnol. 37, 1452-1457. DOI 10.1038/S41587019029-0. Chen, W. Guillaume T. O. Dionysi, R. Reiner, P. Y. Zachara, M. Gabeline, C. G. et al. 2021. Genome-wide molecular recording using LifeSec. BioArchive 03, 436,752. DOI 10.110.12021.03.24.436752. Denise A. Richter, M. L. Fisher, D. S. TSF, J. and Column Tach, M. 2019. EpiScanB, Integrated Single-Cell Epigenomic Analysis, BioArchive. DOI 10.1101648,097. How, Y, How, S. Anderson Nissen, E. Mock, W. M. Zhang, S. Butler, A. et al. 2021. Integrated Analysis of Multimodal Single-Cell Data. Cell 184, 3573 to 3587. DOI 10.1016/J.Cell.2021.04.048. Lamano, G. Soldatov, R. Seisel, A. Braun, E. Hotchkerner, H. Petahov, V. et al. 2018. RNA velocity of single cells. Nature 560, 494 to 498. DOI 10.1038/S4158601804114-6. Labib, M. and Kelly, S.O. 2020. Single cell analysis targeting the proteome, NAT. Reverend Chem. 4, 143 to 158. DOI 10.1038/S41570200162-7. Lonman, D. Custer, J. Shurek, E. McCarthy, D. J. Hicks, S.C. Robinson, M.D. et al. 2020. 11 grand challenges in single cell data science. Genome Biol. DOI 10.1186/S13059902926-6. Ledith, O. Rod, J. Lang, S. Dapla, P. Polson, J. Wallegard, M. et al. 2019. singlecellVR, a virtual reality platform to visualize and analyze single cell data. BioArchive. DOI 10.1101329102. Luo, C. Haikova, P. and Ecker, J. R. 2018. Dynamic DNA methylation in the right place at the right time. Science 361, 1336 to 1340. DOI 10.1126/Science.at6806. Ma, S. Zhang, B. Lefebvre, L. M. Earl, A. S. Cheng, Z. Hu, Y. et al. 2020. Chromatin potential identified by shared single cell profiling of RNA and chromatin. Cell 183, 1103 to 1116. DOI 10.1016/J.Cell.2020.09.056. Miller, B. F. Bamba Muka, D. Dulac, C. Zhuang, X. and Fan, J. 2021. Characterizing spatial gene expression heterogeneity in spatially resolved single-cell transcriptomic data with non-uniform cellular densities. Genome reads 271,288, 120. DOI 10.1101/GR.27128.8.120. Nestaroa, S. Hamey, F.K. Poinsala, B. Diamante, E. Shepard, M. Lorente, E. et al. 2016. A single-cell resolution map of mouse hematopoietic stem and progenitor cell differentiation. Blood 128, E20E31. DOI 10.1182/Blood 2016-05716480. Paul, F. Arkin, Y. A. Gillidy, A. Jayton, D. A. Koenigsberg, E. Karen Schall, H. et al. 2015. Transcriptional heterogeneity and lineage commitment in myeloid progenitors. Cell 163, 1663 to 1677. DOI 10.1016/J.Cell.2015.11.013. Perkel, J. M. 2021. Single cell analysis enters a multiomics age. Nature 595, 614 to 616. DOI 10.1038/D4158602101994W. Sellens, W. Knoet, R. Todorov, H. and Sayes, Y. 2018. A comparison of single cell trajectory inference methods towards more accurate and robust tools. BioRexiv. DOI 10.110127600907. Shom, N. Carcanius, J. Neff, N. F. May, AP, Quake, S. R. Wiskorai, T. et al. 2018. Single cell transcriptomics of 20 mouse organs creates a tabula muris. Nature 562, 367 to 372. DOI 10.1038/S415866018-0590-4. Spark, A. Kitching, A. Esteban Ferrer, D. Handa, A. Carr, A. R. Needham, L. M. et al. 2020. Bloom, 3D virtual reality for single molecule localization microscopy. NAT. Method 17, 1097 to 1099. DOI 10.1038/S41592000962-1. Spect, H. Emmett, E. Petelski, A. A. Huffman, R. G. Perlman, D. H. Sarah, M. et al. 2019. Single cell proteomic and transcriptomic analysis of macrophage heterogeneity. BioRexiv. DOI 10.1101665307. Stefani, C. Lacey Holbert, A. and Stillman, T. 2018. Confocal VR, immersive visualization for confocal microscopy. J. Mole, Biol. 430, 4028 to 4035. DOI 10.1016/J.J.M.B.2018.06.035. Stewart, T. Butler, A. Hoffman, P. Halfmeister, C. Papalexi, E. Mock, W. M. et al. 2019. Comprehensive integration of single cell data. Cell 177, 1888 to 1902. DOI 10.1016/J.Cell.2019.05.031. Stewart, T. and Satija, R. 2019. Integrative single cell analysis. NAT. Reverend Janet. 2257 to 272. DOI 10.1038/S4157601900993-7. Swanson, E. Lord, C. Reading, J. Hubeck, A. T. Genge, P. C. Thompson, Z. et al. 2021. Simultaneous trimodal single cell measurement of transcripts, epitopes, and chromatin accessibility using TSEC. eLife 10. DOI 10.7554/eLife.63632. Tian, L. Dong, X. Freytag, S. Lei Kao, K. A. Su, S. Jalal Abadi, A. et al. 2019. Benchmarking single-cell RNA sequencing analysis pipelines using mixture control experiments, NAT. Method 16, 479-487. DOI, 10.1038/S4159019, 0425-8. Trapnell, C, 2015. Defining cell types and states with single-cell genomics. Genome Rees. 25, 1491-1498. DOI, 10.1101/gr.190595.115. Wang, Y, Li, Q, Lu, L, Zhou, Z, Ruan, Z, Kong, L, et al., 2019. TerraVR empowers precise reconstruction of complete 3D neuronal morphology in the whole brain. Nat, come on. 10, 1-9. DOI, 10.1038/S41467-019 to 11,443 year. Weinrib, C, Woolach, S, 2C, B, K, Sokolowski, M, and Klein, AM, 2018. Fundamental limits on dynamic inference from single-cell snapshots. Proc, National, ACAD, Sci. USA 115, E2467, E2476. DOI, 10.1073/Penis.17147231115. Welch, J, D, Kozareva, V, Ferreira, A, Vanderberg, C, Martin, C, and Micosco, E, Z, 2019. Single-cell multi-omic integration comparison contrast features of brain cell identity. Cell 177, 1873-1887. DOI, 10.1016/J.Cell.2019.05.006. Wolf, F.A., Angerer, P., and Theas, F., J., 2018. Scampi, large-scale single-cell gene expression data analysis. Genome Biol, 19. DOI, 10.1186/S130590117, 1382-0. Wolf, F.A., Hamey, F.K., Plas, M., Solana, J., Dolan, J. S., Gotchins, B. et al., 2019. Paga, graph abstraction reconciles clustering with trajectory inference through a topology-preserving map of single cells. Genome Biol, 20. DOI, 10.1186/S130590191, 1663X. Singh, Q, R, Farron, C, A, E, Zhang, Y, Y, Yi, Y, Warrior, T, Gautam, P. et al. 2020. Parallel bimodal single-cell sequencing of transcriptome and chromatin accessibility. Genome Rees. 30, 1027-1039. DOI, 10.1101/GR.257840.119. Yang, A, Yao, Y, Li, J, and Ho, J, W, K, 2018. StarMap, immersive visualization of single-cell data using smartphone-enabled virtual reality. BioRuxiv. DOI, 10.1101324524,855. Yao, Z, Win, T, N, Van Velthoven, C, T, J, Goldie, J, Sedino Cortez, A, E, Baftazad, F, et al., 2020. A taxonomy of transcriptomic cell types across the isocortex and hippocampal formation. BioRuxiv. DOI, 10.110120.03.30.0152214. Ju, C, U, M, Huang, H, Jurek, I, Abnesi, A, Hu, R, et al., 2019. An ultra-high throughput method for single-cell joint analysis of open chromatin and transcriptome. Nat, Struct, Mol, Biol. 26, 1063-1070. DOI, 10.1038/S415940190, 0323X. Keywords, single-cell, Skrinosec, Scaticsec, virtual reality, VR, data visualization, clustering, trajectory inference. Citation, Stein DF, Chen H, Vinyard Ni, Chin Q, Combs RD, Zhang Q, and Pinello L, 2021. Single-cellular, interactive visualization of single-cell data in virtual reality. Front, Genet, 12-764170. DOI, 10.3389/FGene.2021.764170. Received, August 25, 2021. Accepted, September 27, 2021. Published, October 28, 2021. Edited by Guangzhuang Yu, Southern Medical University, China. Reviewed by Dake Zhang, Beihang University, China. Indu Country, Leiden University Medical Center, Netherlands. Copyright 2021, Stein, Chen, Vinyard, Chin, Combs, Zhang, and Pinello. This is an open access article distributed under the terms of the Creative Commons Attribution License, CC BY. The use, distribution, or reproduction in other forms is permitted, provided the original authors and the copyright owners are credited, and that the original publication in this journal is cited, in accordance with accepted academic practice. No use, distribution, or reproduction is permitted which does not comply with these terms. Correspondence, Luca Pinello, Pinello at mgh.harvard.edu.